Okay, are we ready? All right, let's do this. So many predators have evolved unique specializations to take advantage of at least spending time in the mesopelagic. So as an example of that, swordfish spend all day around 500 or 600 meters. I'll show you that again in a minute. But they've evolved, large eyes. They have a heater in their brain, so their visual acuity is much faster than their prey at depth. They happen to have a sword on their face. So they're a really sort of formidable mesopelagic predator, and I think therefore I would argue that these things have evolved with this biomass in the deep ocean, and we could actually use them equipped with sensors like you see here to help us make observations in this place that is really difficult to study, and I don't need to tell you guys that. So sticking with that swordfish example for a second, just to convince you, if I haven't yet, that these guys are really great ocean observing platforms. So this is just an example from one of the tags we put on a swordfish. This is about 6 months of data going across on the X, and their depth temperature profiles on the Y-axis. Of course, color is temperature here. And so what should jump out at you is that every single day, which are these little vertical bars, this animal moves from the surface to about 500 meters like clockwork, day after day after day. I'm going to get into a little bit some of these anomalies you see here. I'm sure that jumps out at you too, but the take home from this is just that these things go there a lot, and so we can use that to our advantage. It turns out that we, as a research group here at WHOI, but also we as a community, have tagged a whole bunch of predators with various sensors, and it turns out that pretty much all of them use the deep ocean, twilight zone, even into the midnight zone, from swordfish to whale sharks to white sharks, mako sharks, seabirds even, leatherback turtles. This is a figure from a review paper we did last year that simply looked at across predator guilds, what is sort of the habitat use like in the vertical? And what you can see here is that in many cases, these animals are using the mesopelagic and even into the bathypelagic. So, there's got to be something driving the kind of convergent evolution of the specializations that allow these animals to use this habitat that is otherwise, high pressure, cold, dark, very inhospitable. So this is not my work. This is from Yoshino et al. A couple of years ago. This is from a head-mounted camera on an elephant seal. And so I'll run that back for you one more time. This is why elephant seals are going to the mesopelagic. And here for a squid. So for some taxa, such as elephant seals, also beaked whales, foraging is a really clear driver of mesopelagic habitat use. The evidence is unequivocal, as you can see here. And I hate to end this, but it ends the same as it did for the fish. Spoiler. So that's kind of, there's a few examples in that space of how predators use vertical habitat where we have unequivocal data that proves to us that this habitat use is indeed for foraging. In contrast to that, if I jump back to the swordfish for a second, on the left-hand side, you're going to see what I've been alluding to now, which is a very typical, 24 hours or so of, a day in the life of a swordfish, basically. And what you see is during the daytime, the fish is down around 500 meters, and the nighttime, some of which is cut off here, they're in the epipelagic, and it's like clockwork. So you can see sunset, the fish descends, and sunrise, the fish descends, sunset, back to the surface, and every day like clockwork. So you might look at that and think, okay, it's at least tracking DVM as we know it, right? And so maybe that is also indicative of foraging. I'm maybe not convinced, but it's possible, right? On the right-hand side, what you see is, I would suggest, is absolutely not foraging behavior, at least in the beginning. So you see the fish makes this really rapid descent to about 1,500 meters, comes back up well above the primary DSL, and then eventually kind of levels out into that normal behavior. So this is one example of many. If you want to see more, you can check out this review paper, but there are many, many examples where use of the mesopelagic and the bathypelagic are behaviors that I would suggest are really not indicative of foraging. And so there's sort of this open question about the functional role of these animals using the deep ocean. But again, there's this selection pressure that's driving them to use it, right? So there's good reason for them to go there, but we don't really know what that reason is. And so this, of course, leads to this very obvious question. That was in review space, what I was just talking to you about, which, as you guys know, is sort of often qualitative. And frankly, the answers to those questions about the functional role are sort of unsatisfying in that space, except for a few of the good examples. So we sort of set out to really try to test this question with some of our own data. So we aggregated a bunch of animal tag data from our work and from a bunch of our colleagues throughout the North Atlantic. This represents about 12 different species, about 350 individuals, 42,000 days of 3D movement data, these predators moving up and down between the epi and mesopelagic. And I think, as I'm showing you the kind of their general movements over there on the left, it's something like 1.5 million kilometers or something that they traverse in the North Atlantic. So it's like a really cool data set from an ocean observing perspective. And as you might have guessed, sort of following from the results of that review paper, when we look across all those species and we just kind of pool them all together, what we see is basically every grid cell that has a color in it suggests that the mean across species puts the daily depth in the mesopelagic. And you can see there's effectively no gray squares, which would suggest no use of the mesopelagic. So across the basin, across the board in these taxa, they're all going there, they're all spending time there. And so we kind of confirmed with our own data, okay, this is a real signal that we're seeing that the mesopelagic is indeed an important habitat. So that was fine, that's not really what we set out to do. What we set out to do is test this question about foraging. And so Martini actually built this modeling framework that looks at the relative impact of biotic and abiotic drivers on the daytime, we call it the Q95 depth, so the 95% quantile close to the max, to try and really get at what's driving this deep diving behavior. And so it was relatively simple the way we set up these drivers. So for the abiotic, we just used water column thermal structure as a primary driver / modulator of behavior. And then our biotic driver was the predicted depth of the primary DSL. And we can talk about that later if you want to. But we don't have, synoptic measurements of the DSL. And so we had to predict that, and we pooled some model results together to result in this kind of synoptic prediction. Happy to talk about that more. But at the end of the day, we sort of bring these things together to try to predict vertical habitat use of these predators. This is, I promised, the sort of media slide I have, so I'm going to walk you through it. So we have this same slide for 12 species, but it's super overwhelming. I'll show it to you later if you want to see it, but I just grabbed 3 examples that represent what I think is kind of the spectrum. So to orient you, the y-axis here is showing you the actual observed Q95, that sort of near max depth that these predators are using every single day. The x-axis is what the model predicts. And so for blue marlin, on the left-hand side, you can see the model's not really capturing the variability that you see in the observations. But as you move to the right, for blue sharks and for swordfish, you can see there is actually a pretty good relationship. Now, there's a lot of variability. There are plenty of caveats associated with this, but I think given all those caveats, we still see this relationship. I think we're pretty happy with the way this looks. And so then we ask ourselves, well, what's the interpretation? And this is where it gets really interesting. So to orient you again, the y-axis now, you're looking at the DSL depth in a place on a day. And the x-axis is showing you the thermal structure of the water column represented by a temperature gradient, vertical temperature gradient. We built this metric that just shows you basically as the reds get more red, the animals are above the predicted DSL depth, and the same is true for blue, they're below the DSL depth. The light colors show you they're sort of closer to that DSL. And so the interpretation of this for the blue marlin, it's really dark red. It doesn't really change across the X or Y gradient. And our conclusion there is pretty much that they're always above the DSL. They seem kind of disconnected from some of these drivers that we're using. In contrast, if you jump to the right for the swordfish, you see these really light colors, which suggest that they're close to the DSL, and they're always blue, which suggests they're right below the DSL pretty much all the time. And it doesn't matter what the temperature gradient is or what the DSL depth is, right? They're tracking that sort of across this temperature and DSL space. An intermediate case is the blue shark, where you see really light colors on the right-hand side where the temperature gradient is low. suggesting that they're pretty tied to DSL depths in that case, but as you get a stronger thermal gradient at depth, they become disconnected from the DSL, and that sort of aligns with what we know about their thermal physiology and sort of limitations to diving. So we kind of expected some of these results, but it was really neat to be able to pull all this data together at a basin scale across dozens of individuals and a dozen species and actually see these patterns manifest. So we have some sense across those 12 species, which ones maybe are more connected to the DSL and which ones are less, sort of pointing at least indirectly to the potential foraging role for some of this vertical habitat use. But we weren't super satisfied with that high level result as a way to sort of disentangle this complexity. And so we took that big data set of predators and we matched it to concurrent shipboard acoustic data. Now that's, for those of you who have worked with that data and maybe also with tag data, you can imagine that's sort of a Herculean effort, and there's really not that much concurrent predator ship data for a number of reasons. But to sort of just dig into that a little bit, we did pull out some case study examples that I really love because they highlight the really the complex nature of these relationships and how sort of detailed you can get and granular about these potential predator-prey interactions and how these systems sort of structure each other. So the most simple example is the swordfish. Again, you can probably could have drawn this with a crayon if I'd asked you to, but basically the shipboard acoustics are underneath where those darker purples are more backscatter. And what you see is the swordfish is, as I sort of alluded to before, tracking the DVM like clockwork, right? You could set your watch to when that fish descends and then ascends again, really closely tracking the scattering layers. So the swordfish is over the color in the black, as you figured out. The white points are actually tag detected bioluminescence events. The tag has a light sensor, and so that's how we calculated that. The presence only metric, but what you can see is you see a couple of bioluminescence events there in that morning crepuscular period, indicating that at least the predator is interacting with some kind of bioluminescent scattering layer organisms. The predator-prey interaction, of course, is still unknown, but that at least gives us some confidence in sort of doing these overlaps. This one's a little bit more complicated, but maybe even more interesting. So we saw for the blue shark that there's some disconnect between the predator's behavior and DSL depth in some cases, but they also tended to be tied to them in some cases as well. And what we see here in this particular example is, again, that bioluminescence is in the white, which really should jump out at you. I've labeled here, of course, the DSL and a couple of shallow scattering layers. That first one looks to be non-migratory, and then there is a second one kind of under all the shark data. It's a little bit harder to see. And in some cases, maybe you wouldn't notice it in the acoustics, except that we have bioluminescence on the tag saying there's a lot of bioluminescing organisms in this layer where the shark is hanging out. So again, you can start to think about, what is really driving these interactions at super fine scales. And this is something we're super excited about. And just talking about shallow scattering layers, if you saw Martini's lightning talk yesterday, he's been doing some cool work actually looking at the energetics of predators interacting with the scattering layers, in this case in the North Pacific subtropical gyre. And he has some good evidence that suggests that this is sort of how those predators are making a living in the open ocean by interacting with and taking advantage of that shallow scattering layer. So many of you know shallow scattering layers are something that we don't really have a good understanding of. They tend to be really patchy, but it's something that it seems like the predators know about and are taking advantage of. So his paper's coming out soon. Check that out or ask him about that work. So that was by far the longest part, I promise. But the sort of a summary for part one is just that I hope I've convinced you that all predator taxa are using the deep ocean. It's an important habitat for them. The behavior suggests that while some are clearly motivated by foraging in the deep ocean, there are clearly diverse motivations for deep diving. And as those acoustics examples I hope highlighted for you, it really requires these kind of multidisciplinary approaches, bringing together, for example, tags and acoustics. Those are really required to understand how these relationships work, and of course to make observations in these places that are difficult to study. This is the black mesopelagic slide. So I just want to say that hopefully that gives you a sense for the potential power of using these animals as ocean observing platforms, especially in the deep ocean. So Nella's coming up next. I know she thinks a lot about combining sort of complementary approaches for these difficult observing problems. I think you guys saw a lightning talk from Elon yesterday. He's going to have a poster tonight talking about how predators can be used as sampling platforms, which I'm super excited about. Obviously, I'm biased, but check that out. And so transitioning then from predators as observing platforms to kind of what are we doing next? What are we working on right now? Our questions are really about where are the predator-prey interactions occurring? Is it at night when all the prey is coming up into the near surface and they're sort of condensed? Is it during the daytime where predators like swordfish have significant advantages over their prey potentially? Or maybe it's in those crepuscular windows where the prey are more vulnerable. These are all open questions that we're actively working on, and of course have implications for how carbon's being moved around, for example. So that's some stuff we're working on now, and I'll just finish with a little kind of vignette on how we've been dealing with that. So last summer, we partnered with the fishing vessel Monica. It's a commercial longline boat that fishes out of Barnegat Light, New Jersey. We can talk about this more if you're interested, but I'll tell you that working directly with fishers who spend every day on the water, can be our sort of eyes and ears out there, has really changed the game for how we do our science. They've given us access to places and animals that we just otherwise can't touch as scientists. And I would also say that, sorry, I got my slides mixed up. Anyway, I had a nice thing about fishers, but I'll come back to that. In addition to the tagging I'm about to tell you about on the Monica, I also just want to mention in this sort of complementary approaches space, Kira Willis has a poster, I think it's tonight as well, that she is analyzing some of the gut content and isotope data related to this tagging data that was also collected on the Monica. So we sort of got all this data concurrent in approximately the same time and place. So if you want to see her poster, go check that out. And that's sort of in the context of the tag data I'm about to show you. And if you think kids get excited at Christmas for toys or presents or whatever, take a box of tags like this to a professional fisher or commercial fishing boat and let them put the tags on the animals that they spend every day trying to catch and that they're really keen on understanding. I tell you right now, it can't touch kids at Christmas. It's really amazing. I think a few of us can attest to how excited they get to sort of have these new tools at their disposal to understand and sustainably manage their livelihoods, right? It's just really, really cool. So indeed, we took this giant box of tags on the boat, and I'll draw your attention to the top right corner. Those are some biologgers, are sort of like a Fitbit for predators that we built in the lab, and they basically give us things like tail beat frequency and 3D movements. We can sort of fly our way through how an animal uses its habitat in really high resolution. And so the way this works, they're about the size of a cell phone, as you can see on the left there. Thanks, Rodney. But we put these on predators. See a tuna here and a swordfish on the right. After tag and release, they seem pretty happy and have a good story to tell, probably. So the way this works is these little cell phone sized tags, sorry, I should have brought one. We put them out on an animal for a couple of days, maybe in the open ocean, and then we have to go get it back. And so this is a photo on the nicest day when we were out there, where I actually jumped in the water, took this picture just to give you a sense of scale for this tiny tag floating next to, this is like a 70-foot fishing vessel, 200 miles offshore. And so this is, obviously this is gorgeous weather, so it's not that big of a deal, but when the wind blows and there's big waves and whatever, this becomes much, much, much harder. And we've just started now to kind of dissect what this data is telling us. So this is about 170-pound big eye tuna. It's a pretty good size adult fish. And what you'll see in the top left there is those kind of depth temperature profiles, exactly like I've been showing you before, giving you sort of the quintessential big eye tuna behavior during the day. They're making tons of sort of oscillatory dives down into the primary deep scattering layer, maybe just above it. But now what we've added on top of that really high resolution vertical data is, for example, the pitch of the animal in the red. So you're seeing how the animal's moving in this axis. There are other axes of acceleration, but I just pulled out one more here in the green. is a metric of energetics, of how the animal's sort of moving and how that, that movement altogether results in how the animal is burning energy, essentially. So you can kind of with your eye align, you know, a rapid deep dive with a spike in energetics, for example. And one more example on the swordfish. I have to leave you with the swordfish, obviously. Again, you're seeing the kind of quintessential behavior that I've been telling you about, where as the sun rises, the animal descends to depth. That's around something like 500 meters. Again, you see the pitch of the animal is really variable as it's sort of moving through its environment. But instead of energetics now, I've replaced here with the blue line, the role of the animal. And what might jump out at you is there's clearly a step change, right, at that crepuscular period. And these numbers maybe are hard for you to see, but all of this is between 45 and 45. So what the animal's doing is when it gets to depth, it's swimming on its side for a little bit and then rolling and switching to the other side and swimming. We've only measured that, we as a community, have only measured that in one other predator, which is the hammerhead. And that's known to be an energetically efficient way to swim for that animal. And so now we just measured that in the swordfish as well. So this begs the question. I would have guessed, yeah, maybe swordfish are using the mesopelagic to forage, but this would suggest there's at least some energetic component to that deep habitat use where it's super cold, maybe slows down their metabolism, and they're exhibiting this unique swimming behavior that suggests that they're trying to save energy while they're down there. And of course, cameras. So we have a few of these animals tagged with, to have these high resolution data sets, also with cameras, mostly because it's fun, but also because it allows you to see what the animal sees. So now you're on board a swordfish as it's swimming around and collecting this data I just showed you, but also collecting this video footage. And of course, while it's neat to kind of be on board one of these predators, this also actually really helps us validate the behaviors we think we see in that high resolution behavior data with what the animal is actually doing, right? So we can sort of start to ground truth some of these behaviors. So with that, I'll just say, you know, new technology like these tags is helping us uncover some of these behaviors at depth. We're still working on it. These predator-prey interactions remain, for me, a really big question, and I think that results in a lot of unquantified carbon transfer. In fact, I would argue we don't even know the sign on that transfer with respect to predators. And so therefore, the role of predators in the carbon pump remains a key question, and I hope to talk about that more in some of our workshop discussions. Plenty of folks to thank. I won't go through them all, but thank you all for making this possible, and take questions if I have time. Thanks.
Okay,
you have time for questions?
I think we're going to hold the questions for now with the delay, and we'll just do that and find Cam later and ask him questions. So next up is Dr. Anela Choi, who's an associate professor of oceanography at Scripps and one of the foremost experts in deep sea community and food web structure and architecture. And I can say personally that I'm a huge fan of her and her lab's work, and every single time a paper comes out, I have to read it, especially when it's about the jelly web that we almost never get to see until her work started to elucidate that.